Hi, and shall we start?
Yeah, I'm gonna.
Hello, everybody. I'm Avram Hershko. I thought that maybe I'll give you a little history of how the ubiquitin system was discovered because it's instructive for young people to hear how discoveries are made. So I am working now for a long time on the problem of how proteins are degraded in cells. And that was a question that did not interest too many scientists when I started out. When I started out, it was after the times of the great discoveries of molecular biology. Can I have the first slide, please?
This is the first slide. Yes.
I can see it.
1 second. Let me share.
Okay, great. So it became after the great discoveries on the structure of the DNA in the 1950s and the cracking of the genetic code in the 1960s, it became apparent that the gene is a piece of DNA that is the blueprint for the formation of the protein via an intermediary blueprint, the RNA. And because of that, there was a great excitement and justified by excitement, a lot of interest in the problem of how proteins are synthesized using the sequence of a basis in the DNA, and how is the synthesis of specific proteins is turned on and off. So there was a lot of interest in protein synthesis, but because of that, not many people were interested in the opposite process of the degradation of proteins back to amino acids, even though a few thought that it is a basically important process because you can regulate the levels of cellular proteins not only by protein synthesis, but also by protein degradation. So this was the knowledge when I came to the lab of Gordon Tompkins as a young postdoctoral fellow. And at that time, he was interested in how steroid hormones cause the increased synthesis of some enzymes in the liver. And when I got there, I saw it was a large lab, about 20 different postdocs working on different aspects on how a certain protein, tyrosine aminotransferase, is the synthesis is increased by steroid hormones. I saw that was too crowded to do what everybody else is doing, so I asked Gordon Tompkins, can you give me another project? And he said, why don't you study the degradation of the same enzyme, because it may be also interesting. So that was how I got involved in the problem of protein degradation, on which I am working ever since. And the next slide, can I have the next slide, please? Shows one of the first experiments that I did in the lab of Tompkins as a postdoc. And I did it quite by accident. There was some other reason for it, but what I found was that the addition of potassium fluoride, an inhibitor of energy production, stopped the degradation of this enzyme TAT whenever it was added. So it wasn't what I did the experiment for, but I was very deeply impressed by these results because it showed that energy is needed for protein degradation. Now, why would energy be needed for protein degradation? At that time, it was known that energy is needed only for the formation of a peptide bond, not for the hydrolysis of peptide bond. So I thought that we may have as yet unknown mechanism that uses energy for the high selectivity of protein degradation. And that was the question was, what is that new system, energy-dependent protein degradation? And to find an answer, I used biochemistry to set up a cell-free system that faithfully reconstitutes energy-dependent protein degradation, and then to isolate the components of this system. And our first step in this direction was shown in the next slide. Next slide, please. So here we used a system from reticulocyte lysates in which you can see the degradation of a protein, in this case denatured globule, is dependent on ATP, or the source of energy. You can see there is a big stimulation by ATP of the degradation. So we had a cell-free system that recapitulated energy-dependent protein degradation, and we began to try to isolate the components, and that was the first step. We just separated the components on a cellulose anion exchanger, and we got fraction 1 that contained all the proteins that did not bind to the resin, and fraction 2 that all proteins that bound to the anion exchanger and were eluted with high salt. And see there was no degradation at all in fraction 1. In fraction 2 there was a little bit of degradation, but most of the ATP-dependent degradation was lost. And then we found that the addition back of fraction 1 to fraction 2 completely reconstituted ATP-dependent protein degradation. That experiment was done by Aaron Chachanover, who was then a graduate student in my lab and later shared the Nobel Prize with me because of this experiment. So it was published in a not very prestigious journal, Biophysical Research Communications, but I am still very fond of this little paper because that was the first step in the right direction. So next was the purification, and we purified the active component that stimulates protein degradation in fraction 2 from fraction 1, and we found it to be a small protein that was similar to ubiquitin that was known before, but its function was not known. So we found that ubiquitin is involved in energy-dependent protein degradation, but we didn't know how it works, and that took us 2 years, and 2 years later, we found, and next slide, please, that ubiquitin becomes bound to other proteins in an ATP-dependent process, and that is the original experiment from 1980. Here we took the purified ubiquitin, labeled it with iodine, and incubated it with crude fraction 2 from reticulocytes in the absence, that is the first lane, or presence of ATP. And you can see, and then separated the products on an SDS polyacrylamide gel. You can see that that without ATP, ubiquitin remains free and migrates ahead in this SDS gel. But following incubation with ATP, it became bound. That we were very surprised to find it, but we found it. It became bound to a huge number of high molecular weight proteins. So we saw that fraction 2 contains not only the enzymes, but also some endogenous substrates of the system, and maybe ubiquitin is linked to the substrate. And to find that out, we added an artificial but good substrate, lysozyme, in lanes 3, 4, and 5. And you can see that several new bands showed up that contained both lysozyme and ubiquitin. And we found that these bands are increasing numbers of ubiquitin molecules bound to one molecule of the substrate lysozyme. So based in that, we postulated that the conjugation of the covalent conjugation, because these are covalent bonds resistant to boiling in SDS, that the covalent conjugation of ubiquitin to protein targets them for degradation. And the next slide, please, shows up our original hypothesis, in which we hypothesized that binding of ubiquitin, or APF, as we called it at that time, targets the proteins for degradation. And the lower part shows the complete biochemical pathway that we found by about 10 more years of work. And as you all know, I guess, those in the field and also those that are not in the field, it's now in the textbook, there are 3 enzymes that are involved in the ligation of ubiquitin to proteins, E1, E2, and E3. I won't go into that. And then once ubiquitin is ligated and several ubiquitins are bound to the protein, it's degraded by the 26S proteasome that specifically degrades ubiquitinated proteins. So I won't go into that, but I just want to point out that these discoveries explained why is energy needed for protein degradation, because that was our original question. So energy is needed both for the ligation of ubiquitin to the protein, because that needs the activation of ubiquitin. So that's one step in which energy is needed. And later it was found that the ATP is also needed for the action of 26S proteasome. And it also explained what determines the high specificity and selectivity of ubiquitin-mediated protein degradation. These are the E3 enzymes, the ubiquitin ligases, that catalyze the transfer of ubiquitin-activated ubiquitin from E2 to the substrate, and they are able to specifically recognize substrate with or without, as we'll see, molecular glues, and target them for degradation. And now about 600 different E3 enzymes are known in the human genome, and we need this large number of E3 enzymes to recognize the huge number of targets of ubiquitin-mediated protein degradation. So that was the original work in which we worked out the mechanisms, the biochemical mechanisms of ubiquitin-mediated protein degradation. But then I saw that here we use mostly artificial substrates such as extracellular proteins, and the question was, how is the ubiquitin system degrading a specific cellular protein? And that's how I became interested in the degradation of cyclin in the cell cycle. Next slide, please. I have the next slide, please, Cedric. Okay, back one slide. So cyclin were discovered by Tim Hunt as proteins that are degraded at the end of each mitosis. And later it was found that there are activating subunits of a protein kinases, like cyclin B is an activating subunit of CDK1. And then when it binds to CDK1 and some other cross-translational modifications, it activates the protein kinase. And cyclin B in the early embryonic cell cycles, cyclin B was known to be synthesized in the interface, to bind then to CDK1, activated, and then in mitosis, the active protein kinase activated many events in mitosis. It was also called at that time MPF, or a mitosis promoting factor. And to get out of mitosis, the protein kinase had to be inactivated, and that happened to be due to the rapid degradation of cyclin B at the end of mitosis. So that was an interesting question on an interesting and biologically important molecule, and I thought maybe I should study this. How is cyclin B not degraded in the interface, and how it becomes rapidly degraded at the end of mitosis? And people, including Tim Hunt, thought that there was a cyclin-specific protease, but I thought that maybe it's a ubiquitin ligase, means specific E3 enzymes that recognizes cyclin at the end of mitosis, and the activity of which is regulated, so it is working only at the end of mitosis and not in the interface. So again, I looked for such a ubiquitin ligase in a biochemical way, cell-free system. But for that, I needed a different cell-free system. And here that's how I got to marine biology and to this creature. Next slide, please. That is the common surf clam, Atlantic surf clam. And it has been used at the Marine Biological Laboratory in Woodshof to study cell division. That is because the female clam has a great number of oocytes, hundreds of millions of oocytes, which can be fertilized by the addition of clam sperm. And then all these oocytes begin to divide at a high synchrony in the early embryonic cycles. And it was known that it's possible to make extract from them that faithfully reproduce cell cycle stage-dependent degradation of cyclin and other cell cycle stage-dependent processes. So I use this system to try to find out how cyclin is degraded, and is there a cyclin-dependent, cyclin-specific ubiquitin ligase. Next slide, please. And in 1995, we found a large complex that we call the cyclosome that contains cyclin selective ubiquitin ligase activities. It was inactive in the interface, became active in ubiquitin ligase, in mitosis, and the activation was due to phosphorylation, this protein kinase, CDK1 cyclin B. So we called it cyclosome because of its large size and its important role in the cell cycle. And the same complex was discovered at around the same time by the group of Mark Kirchner. They called it anaphase promoting complex, APC, and now it is called by these two names, anaphase promoting complex cyclosome, or APCC. Next slide, please. In a paper in TIPS, 1996, I proposed the following model for the regulation of APCC and in the degradation of cyclin. So the APCC is inactive in the interface. It becomes active by phosphorylation by the active CDK1 protein kinase, CDK1 cyclin B. So phosphorylation turns it to the active form, and then the active form targets cyclin B for ubiquitination, and thus targets it for degradation by the proteasome. And then afterwards, the APCC turns back to the inactive, unphosphorylated form by the action of a phosphatase. So the main idea here is the interrelationship between protein phosphorylation and protein degradation in cell cycle control. The protein kinase activates the ubiquitin ligase, and then the ubiquitin ligase inactivates the protein kinase and stops its action. And that interrelationship between protein degradation and protein phosphorylation is a recurring term in many types of cell cycle regulation, also in other regulatory events. Next slide, please. That was the beginning, and the APCC became a field in which many investigators were interested. It was found that it is conserved in evolution from yeast to tramp to man. It's the same APCC almost. It also has several other important substrates besides mitotic cyclins. And here is one important substrate, securin. Securin is an inhibitor, of a protease called separase. Separase is the protease that cleaves the sister chromatid glue between them, the cohesin complex, and but it can be activated, or it is active only when securin, when it is not bound to securin. So at the end of mitosis, APCC is activated. It targets securin for degradation, and therefore separase is now free, and it can now start anaphase by cleaving the cohesin complex. So that is the linkage between the activity of APCC and the initiation of anaphase, the degradation of securin. And that is also how an important checkpoint system works in the cell, the mitotic or spindle assembly checkpoint. That is a checkpoint system that checks whether all chromosomes are correctly attached to both poles of the mitotic spindle, and it acts by inhibiting APCC as long as not all chromosomes are correctly attacked. So that's an important system. And in recent years, I'm working on the biochemical mechanisms of the mitotic checkpoint system, but I won't have time to talk about this work. Next slide, please. And the ubiquitin-mediated protein degradation was found to be important also in other phases of cell cycle control. And here is, besides APCC on the right, you can see another important complex, the SCF skip2 complex. And I worked on this in collaboration with Michele Pagano for several years. And we found that SCF skip2 is the ubiquitin ligase that targets for degradation an important cell cycle regulatory protein, P27. It has also other substrates. So P27 is an inhibitor of another cyclin-dependent kinase, CDK2 cyclin E, which is needed for the initiation of the S phase after the G1 phase. So in quiescent cells, the level of P27 is high, and there it inhibits CDK2 cyclin E. But when cells are stimulated to grow, SCF-skip2 activity increases. It targets P27 for degradation, and CDK2 cyclin E can now start the cell cycle. So protein degradation is important in the regulation of several stages of the control of cell division. And that was how we worked for a while on the role of in cell cycle control, but then many other people got interested, and it turned out the ubiquitin system has a lot of basically important roles. Some of these are shown in the next slide. Besides the control of cell division, it is needed. And I'm just showing some examples in the transduction of certain signals, such as the also in the regulation of certain genes, such as those involved in inflammation, in immunity. Several stages in the development of the early embryo requires selective ubiquitin-mediated protein degradation. It is needed both in apoptotic and pro-apoptotic and anti-apoptotic processes, circadian clocks. So protein degradation is involved in almost every basic process, and that happens by the selective program degradation of either positive or negative cell cycle regulators. Positive, like the cyclin B that I talked about, and negative, like the P27 that I talked about. And also ubiquitin-mediated protein degradation is needed for protein quality control to remove abnormal proteins for cells. So when I started out, I started because I believed that protein degradation is important in cellular regulation, but even I was surprised how important it turned to be. It is almost as important, or maybe as important as protein synthesis in the regulation of basic cellular processes. And because the ubiquitin system is involved in so many processes, it's not surprising that abnormalities or aberrations of ubiquitin-mediated protein degradation causes disease. And the next slide shows some examples, such as cancer. Many types of cancer, abnormality in ubiquitin system is involved. I talk more about that. In some neurodegenerative system, there is accumulation of certain proteins in neurons that may be involved in their damage. So the ubiquitin system may be involved here. In muscle wasting or cachexia, that happens in many terminal diseases, there is muscle, which is, at least in part, is due to rapid degradation of muscle proteins. And that is just some examples of the many important roles of ubiquitin-mediated protein degradation in diseases. And the next slide shows how the ubiquitin system is involved in cancer. As you may know, cancer is either due, or many times is due, to the increase in oncoproteins that simulate cell division, or to the decrease of tumor suppressor proteins that are inhibiting cell division. But you may not know that many oncoproteins, also tumor suppressor proteins, are in a state of rapid turnover. So they are rapidly synthesized and degraded. So when an oncoprotein is synthesized but not degraded, or there's this decreased degradation, the level of the oncoprotein goes up, and it can stimulate abnormal cell division. Similarly, when the tumor suppressor is increasingly degraded, when it's too fast degraded, cell division is not braked as it should be, and cancer ensues. So cancer can be caused either by lack of degradation of oncoprotein or by the too rapid degradation of a tumor suppressor protein. And the next slide shows some examples of that in human cancers. As you may know, colon cancer is many times due to the mutation of APC, adenomatous polyposis coli. And APC normally is involved in the degradation of beta-catenin, an oncoprotein. So when the APC is mutated, beta-catenin is not degraded, levels of this oncoprotein goes up, and that seems to be involved in the pathogenesis of colon cancer, also some melanoma. That is decreased degradation of an oncoprotein. Cervical cancer, as you know, is caused by human papillomavirus, and here the mechanism is that there is a protein E6 that activates E6AP, as we heard from Len. She was the first. She saw the structure of E6AP, and it is known that E6AP targets the P53 tumor suppressor for rapid degradation. And normally, the P53 is targeted by another ubiquitin ligase, MDM2, and which is overexpressed in sarcomas, melanomas, gliomas, and causes the loss of this important tumor suppressor. Another example is the mutation of the von Hippel-Lindau VHL protein. It causes renal cell carcinoma, also some hemangioblastomas, and it is a subunit of ubiquitin ligase complex that acts on transcription factors of hypoxia-induced genes. So when it is mutated, these transcription factors are not degraded, and there is an upregulation of hypoxia-inducible genes, which may help the tumor to survive in hypoxic conditions. So here are some examples of how the ubiquitin system is involved in cancer. And because of that, drug discovery companies got interested, and you heard about the first drug. Next slide, please. First drug that was discovered was Velcade or bortezomib. It's an inhibitor of proteasome. It was developed for other reasons. They thought to use it against muscle degradation, but it turned out to be very good or very useful for the treatment of multiple myeloma, bone marrow cancer. It inhibits the proliferation of myeloma cancer cells and promotes their apoptosis. So that was the first proteasome inhibitors. Now there are other proteasome inhibitors, and you should know that it inhibits the proteasome, but it's used in doses which don't inhibit the proteasome completely. Otherwise, other normal proteins also, normal function of proteins also in protein degradation would not work. But for some reason, myeloma cells are especially prone to partial inhibition of the proteasome, and this turned out to be a very useful class of drugs for the treatment of multiple myeloma. It was the first indication that the ubiquitin system may be a good target for drug delivery. So that is in a nutshell the history. Now, what can we learn from this history? Next slide, please. Maybe one lesson is that not to go with the mainstream, because otherwise the big guys will get there before you if you do it in research. As I said, when I started out, there was a lot of interest in protein synthesis, very little interest in protein degradation. But I decided to go with protein degradation, and it turned out to be well, because I saw that it's important and it's not in the mainstream, so I don't have to compete with the big labs that would obviously get there before me. And another lesson was that sometimes accidental observations are important. So if you have a lucky observation, follow it up. Not all accidental observations are important, but some of them are. So think about unexpected accidental observations. In my case, it was the effect of potassium fluoride to inhibit. When I did the experiment, I saw that it would stimulate the degradation of tyrosine aminotransferase. It actually inhibited it, but I thought about it, and I saw that maybe worthwhile to follow up, and that really led me to try to find energy-dependent protein degradation and the ubiquitin system afterwards. And I also think that my story shows that basic research can lead to translation to the bedside. What I did all my life, and even when now, is mostly basic research, trying to find out how things work. But if you follow up your basic research and you do some discovery, eventually it will be translated. I believe in that. And you saw the Velcade. You see the big activity in drug discovery. You see now today's symposium on molecular glues and how we can approach this important issue. So all this is based on the basic discovery, but of course, then you have to do translational research to follow up your basic discovery. So that is my lessons that I learned. I hope to transmit these lessons to the young people in this audience. And last but not least, I would like to thank the people who helped me all these years. Next slide. So in the Technion, these are Dora Ganot, Hannah Heller, Esther Etan, Sarah Elias, and also Judy Hershko, my wife, helped me in the lab for many years. And I had many, many graduate students, including Aaron Chachanover, who was my graduate student during the fractionation, fraction 1, fraction 2, and the discovery of the role of ubiquitin in protein degradation. And because of that, he shared the Nobel Prize with me. But there were many others who worked on, for example, Yuval Rice worked on the first E3 enzyme, first ubiquitin ligase, how it works, and others who worked on the roles of the ubiquitin system in cell cycle control. And I got a lot of collaboration and help. Ernie Rose also shared the Nobel Prize for his role in the initial work. And I want to thank Michele Pagano, who taught me about SCF skip2, P27, and down are the recent graduate students and postdoctoral fellows who work with me now on the biochemical mechanisms of the regulation of APCC by the mitotic checkpoint. So thank you very much for your attention, and I'm really glad to take questions.
Thank you so much, Dr. Hershko, for starting the event with a brilliant talk. So now it's open up to the platform for questions. So this is Dr. Linus Ling, who is coming from Eli Lilly, who is also our SEEDS board member. So Dr. Ling's question is, how does ubiquitin ligase regulate the kinases?
Well, in the case of APCC, it acts on the activating subunit of the protein kinase. So cyclin B is the activating subunit of the protein kinase, and the ubiquitin ligase degrades it, so it inactivates the protein kinase. In the case of P27, it is the opposite. You degrade the inhibitor of protein kinase CDK2 cyclin E, so you cause it to become active by removing its inhibitor. Either an activating subunit or inhibiting subunit, you can either inhibit or activate the protein kinase.
So, Dr. Hershko, my question is a more general one. You know, obviously, this ubiquitous system itself has to be tightly regulated itself. So you showed one case where a protein kinase is regulating that particular ubiquitinate system. How general is that? Is there other mechanism you're aware that to regulate the ubiquitin system itself?
Yes, there are several different mechanisms. For example, some ubiquitin ligases are rapidly degraded by themselves, so their levels are regulated by degradation. It can be regulated by assistant proteins or by proteins that bind to the ubiquitin ligase and can either activate it or inactivate it. APCC itself is intricately regulated by an inhibitor, for example, in the mitotic checkpoint called the MCC, mitotic checkpoint complex. And then it is activated in different phases of the cell cycle by proteins called CDC20 that activates it in mitosis, CDH1 that activates it during the G1. And there are also phosphorylations, as I showed. It is regulated directly by phosphorylation, but then the activator can be removed or inhibited by phosphorylation of either CDC20 or CDH1. So APCC is an excellent example of the multiple layers of regulation in which ubiquitination or the ubiquitin ligase is controlled. Sometimes ubiquitination is controlled by changes in the substrate. For example, P27 becomes, we showed that with Michele, P27 becomes a substrate when it is phosphorylated on a certain threonine site. So that happens many times, that phosphorylation convert the substrate to be a good candidate or to be the substrate for the ubiquitin ligase. So phosphorylation is necessary for converting the substrate. So these are the different mechanisms in which ubiquitination, ubiquitin ligases, and protein degradation can be controlled.
Thank you very much.
Mm-hmm.